So, ladies and gentlemen, you are all welcome to my YouTube channel. Today, we are going to look at the cell membrane structure and function. And in this video, we are going to also look at how substances transport across the cell membrane. So, the anatomy, first we are going to look at the anatomy of the cell membrane. So, the cell membrane is a bilayered and it it exists in between outside the cell and inside the cell. So the outside of the cell is extracellular environment, but the inside the cell membrane is the cytoplasm or intracellular environment. And this cell membrane is made of a phospholipid, and the phospholipid is a lipid that have phosphate group attached to the lipids, and it's what make the cell membrane it what make the cell membrane to be amphipathic in nature. That is to have some components that will interact with water and some components that will not interact with water. So this is it. So in the phospholipid, we have a polar region. That is a region that interacts with water. So it is the head. It is the head part of the phospholipid. And then there is also the tail region. So the tail region, it is the non-polar region of the phospholipid. Like for example, from here to here, it is a tail region. So it is a non-polar component, or it's a non-polar region of the phospholipids, and it doesn't interact with water molecule, while the polar region is a region that interacts with the water. So then the next one is the selectivity permeability nature of the cell membrane. So generally cell membrane allow, permeate means allow. So a characteristic of cell membranes that allow it to regulate the passage of the molecules. So selective permeability nature does not depend on the structure of the cell membrane, and is not actually to be confused with a semi-permeability. So dialysis oven is a semi-permeability. So what characteristics allow a molecules to travel through the cell membrane? So our cell membrane are selective. So our cell membrane are selective due to the presence of two major biomolecules on the cell membrane. So the cell membrane is usually selective. So when we said it's selective, it means that it allows some certain molecules to get into it and also allow some molecules to exit the cell. Also, this allows some materials to get in and exit of the cell. So that is the selective permeability nature of the cell membrane. So the selectivity permeability nature of the cell membrane is due to the presence of the membrane proteins which actually embedded within the cell membrane, and then these proteins decide what can pass, whether it is a big, small, whether it is necessary or unnecessary, whether the molecules is charged or not. And then the next one is the properties of the phospholipids, and that is what make of the layer of the phospholipid. And of course, the properties of this phospholipid is what actually make a non-polar or polar biomolecules, that is non-polar or hydrophobic molecules, to pass, exit, or enter into the cell. So these are the two major biomolecules that make the cell membrane to be selective. And then the next thing, we have a membrane protein. So membrane proteins, we have some of these membrane proteins. We have example, we have transporters, we have anchors, we have receptors, and we have enzymes. So some biomolecules might need help of a protein for them to get through the cell membrane. So some require the help of transport proteins for them to get into the cell. And of course, some of these proteins that are found on the membrane proteins, some of these proteins that are found on the cell membrane include transporter proteins, anchors proteins, receptors proteins, and enzymes. For example, some drugs some microorganisms require receptors before they get into the cell. But generally most of the microorganisms in the body, they are dangerous and we don't need them. But still they need receptors before they get into the cell. Like for example, in the case of human, we have what we call like angiotensin converting enzymes receptor, which is required by the coronavirus to get in. So there's also a drug receptors interaction. Some drugs they can't get into the cell until they interact with their specific receptors. And there are also enzymes that are found in the cell membrane, which is also very, very important for the transport as well as the regulation of what get in and outside of the cell. And of course, we have a transporters proteins. And some of these transporters proteins, we have channel proteins. So the molecules use these transport proteins to get in or out of the cell. So there are some biomolecules that require the need of the transport proteins before they get into the cell. And let's see examples of those proteins. Generally, large molecules require the need of the transporter proteins before they get into the cell. And also charged molecules or ions. Charged molecules or ions also need a transport proteins before they get into the cell. And then we have the molecules that might be traveling off their concentration gradient. That is when you have the molecules moving to the region where their concentration is very high, so they also require the need of transport proteins before they get into that cell. Like for example, if you have two regions and the first region you have 10 molecules of that molecules, and where it's trying to move, maybe there are 100 of them. So they need a transporter protein that will carry them and get them into the region. And then the next one is the cross section of the cell membrane. So which molecules will allowed through the lipid bilayer and which ones may need the help of the proteins. So like carbon dioxide, water, glucose, and starch, we have a lot of glucose, potassium ion, estrogen, and sodium ion. So these are the kind of the molecules that require the help of proteins for them to get into the cell. But why or why not? So now let's just recap briefly on the cell membrane. So the characteristics of the cell membrane, as we said, it is selectively permeable and it regulates what enters and exits cells, and it helps transport certain molecules across with the help of proteins. And the lipid is bilayered, it has two layers, and this layer is hydrophobic and hydrophilic component. And there are embedded proteins, and these embedded proteins have different functions. And some of the functions of these embedded proteins include the transport of molecules, enzymes, cell identification, and etc. So some of the proteins that are found on the cell membrane, some of them they have in the they have for the identification of cells, identification of pathogens, identification of one cell, and identification of not on cells. So all these proteins is some of their functions. So the next thing is actually the terms that are usually interchangeable: cell membrane and plasma membrane. Like whenever you see cell membrane and plasma membrane, they reflect the same thing. And then phospholipid bilayer. When we said phospholipid bilayer, it means the layer of the lipid where there are polar and non-polar components. And then there is only lipid bilayer. So lipid bilayer and phospholipid bilayer are not the same thing. And then the next one is fluid mosaic model. So when we said fluid mosaic model, it means that it's a model that's trying to talk about how nature the membrane is. So generally membrane, it is fluid in nature, and some of the lipids, some of the component of the membrane, and move from one point to another because of the fluid nature of the cell membrane. And then we have types of molecular movement across the cell. We have diffusion, we have transport, passive transport, we have under passive transport, we have simple diffusion, we have facilitated diffusion, we have osmosis, we have active transport, and then, of course, when we said diffusion, it means the movement of molecules from the region of higher concentration to a region of lower concentration. What does that mean? It means that when you have a situation where you have a molecules in a region where their concentration is very low, and of course you have a region where their molecules is very, like for example, now if you have, let's say this is a two region, and of course in the first region you have the molecules like this, you have the molecules like this, these are the molecules, and of course here you have the molecules also like this. So if you have a region where the concentration is higher, like for example, the concentration here of the molecules is higher compared to here, this one is low. So when the molecules is moving from the region of higher concentration to the region of lower concentration, that is diffusion. Then we also have passive transport. So in the case of passive transport, we have simple diffusion and we have facilitated diffusion. So in the case of simple diffusion, it's also a movement of molecules from the region of higher concentration to a region of low concentration. And that is what you call a simple diffusion. And it doesn't require any energy. It doesn't require proteins. Like for example, generally, when we said passive transport is a type of transport that do not require energy for it to transport the molecules. So generally, passive transport do not require the use of any energy for it to transport the molecules across the cell membrane. So under that passive transport, that is where we have simple diffusion and then facilitated diffusion. So when you say simple diffusion is also a movement of molecules from the region of higher concentration, a region of low concentration without the use of energy or protein carrier. And we have facilitated diffusion. So under facilitated diffusion is also a movement of molecules from the region of higher to a lower concentration. But in this case, facilitated diffusion require the use of protein carriers. It requires protein carriers. Then osmosis is a movement of molecules also from the region of higher concentration to a region of low concentration, but it doesn't need or it doesn't require the use of protein carrier. Rather, it requires a semi-permeable membrane. So when molecules are moving from the region of higher concentration to region of low concentration through a semi-permeable membrane, and that is osmosis. In fact, that is what made diffusion and osmosis differ from one another. Osmosis is required to move it through semi-permeable membrane. Then the next one is active transport. So active transport is a transport of molecules from the region of lower concentration to a region of higher concentration. So you see, this is the one that is differ from a low high concentration, and it requires the use of ATP, that is energy by the cell, and it also requires a transport protein. So it also requires transport proteins. So now let's look at the differences between simple diffusion and facilitated diffusion. So in simple diffusion, molecules can easily pass through the cell membrane, and the molecules' characteristics allow it to pass the hydrophobic part of the cell membrane. And of course, diffusion is from passing from the region of higher to a lower concentration. But in the case of facilitated diffusion, also molecules have to pass through a membrane proteins to cross the membrane. So you see, they require a protein channel. There is a channel protein before they get into the cell. So, and in this case, the molecule's characteristics do not allow it to pass through the hydrophobic part of the membrane. And of course, it's also true a region of higher to lower concentration. Then we have what they call endocytosis and exocytosis. So in the case of exocytosis and endocytosis are bulk transport process. So remember, we talk about active transport and passive transport. So endocytosis and exocytosis are also fall under bulk transport. So generally, the types of transportation are categorized into three. We have active transport, we have passive transport, and then lastly, bulk transport. So endocytosis and exocytosis are bulk transport process for moving materials into and out of the cell. So in the case of endocytosis, cells intake external materials by engulfing them with the cell membrane. While in the case of exocytosis, cells accept material by fusing a vesicle with the membrane, releasing the content outside the cell membrane. So that is the differences between the two. So the movement of molecules in the case of endocytosis is by engulfing them. But in the case of exocytosis is by fusing the material with a vesicles and so generally, the large molecules that cannot, or the two large molecules that cannot directly pass through the bilayer of the cell membrane are the one that use these types of transport, that is endocytosis and exocytosis. And examples of these large molecules includes microorganisms and waste products. So are some of the substances that move through the membrane via exocytosis and endocytosis. Also, like for example, hormones that need to be released, they also use these types of transport system. That is endocytosis and exocytosis. And also bacteria, which is one of the examples of the microorganisms to get into the cells by engulfing, and that is also another reason why we have this endocytosis and exocytosis. So, and then the next thing, so that is actually for the basic concept of the endocytosis and exocytosis. And then the next types of the next types of this transport, we are going to look at the active transport very closely. So molecules that pass through a protein to cross membrane. So not diffusion, because active implies a movement of solid against their concentration gradient. That is from the low, the high region where the concentration is high, from the region where the concentration is low, the region where the concentration is high. Being active, it means it requires energy. It requires energy in the form of ATP, and then it requires protein or forms to transport molecules across the cell membrane. So it means that in the case of active transport, there is a need for proteins. There is proteins or pumps, and also there is a need for energy, the transportation of the molecules to take place. So there are two types of active transport. We have primary active transport. So primary active transport uses ATP, while secondary active transport uses an electrochemical gradient. So what is primary active transport? Primary active transport is also called direct active transport because directly it uses metabolic energy to transport molecules across a cell membrane. And examples of the molecules that are transported by this primary active transport are generally ions, like sodium ions, potassium ions, magnesium ion, and calcium ions. So examples of this primary transport, we have sodium potassium pump and we have calcium pump. These are the best examples of the primary active transport. And the transport of charged particles require ion pumps channel. So it means that in the case of primary active transport, it requires ATP and then a protein pump. The secondary active transports. In secondary active transport, also known as coupled transport or co-transport, the secondary active transport, there is no direct coupling of ATP, but it depends often on the electrochemical potential differences created by pumping of ions in and out of the cell. So generally, in the case of the secondary active transports, it doesn't require the coupling of ATP, but usually electrochemical potentials are generated when ions are pumping in and out of the cell. So that electrochemical potential is what have the movement of the molecules. So, and the best example of this is sodium glucose transporters. Because this secondary active transport is divided into three. We have Uniport, Symport, and Antiport. So a uniport, uni means one. It means that when you have and for the uniport, it means that when only one molecule is getting in or out of the cell. And Symport, when you have two molecules moving simultaneously in the same direction across the cell membrane. Like for example, if you look at this, you see that the reds and the blue ball are entering in the same direction. They are moving in the same direction. So that is Symport. But in the case of Antiport, it means that they are the two molecules that are moving simultaneously but in opposite direction. Like for example, look at these balls, they are moving down, and this one, as this one are moving down, and this one are going up. That is, as this one are getting into the cell, so this one are getting out of the cell. So these are the types of transports across the cell membrane. Ladies and gentlemen, if this is the first time you are coming to my YouTube channel, please subscribe. Thank you.